Thomas Hunt Morgan and fruit flies. Who was Thomas Hunt Morgan? Thomas Hunt Morgan was born on September 25, 1866 in Lexington, Kentucky, in the United States, and died on December 4, 1945 in Pasadena, California. He was an American zoologist and geneticist, and is most recognized for his famous experimental research with fruit fly Drosophila, by which he established the chromosomal theory of heredity. He showed that genes are linked in a series of chromosomes and are responsible for identifiable, otherwise known as sex linkage. Morgan's work played a key role in establishing the field of genetics. He received a Nobel Prize for Physiology or Medicine in 1933 due to his excellent work. So, the description of his experiment that proved sex linkage. He found the first useful fruit fly mutation, as flies normally have red eyes, but he found a male with white eyes, so he was a mutant. He immediately crossed the male with a female with red eyes, and all the flies in the first generation, F1, were red-eyed. He then crossed males and females from the F1 generation with each other. The white eye mutation reappeared in the Mendelian ratio 3:1 as a recessive gene. However, only males show this white-eyed phenotype. This suggested that mutation is carried on the X chromosome. Crossing the white-eyed males and red-eyed females from the second generation produced equal numbers of offsprings with each eye color. Males with white eyes have inherited the mutant gene from the X chromosome from their mother, while females only show the trait if they inherit mutant genes from both X chromosomes. This phenomenon is called sex-linked inheritance. So, now, gene linkage maps. Morgan identified that a number of different traits that didn't follow the Mendelian ratio, such as certain phenotypic combinations, occurred in much lower frequencies. He made two key proposals. The alleles for these traits were located on a shared chromosome, otherwise known as gene linkage or sex-linked. The other proposal is that the linked alleles could have uncoupled via crossover to create alternative phenotypic combinations. These discoveries led to the idea that crossover frequencies depend on the distance between two genes, so a gene with a higher frequency are further apart from the genes with lower frequency. He used this concept to develop the first gene linkage maps. So from this diagram, it can be seen that long aristae and red eyes are more likely to separate, therefore they have higher crossover frequencies. However, long aristae and long legs have lower crossover frequencies. This indicates that aristae and leg genes are closer together, while the gene for eye color is further apart. So how does this differ from Mendelian inheritance? Sex linkage. A linkage group is a group of genes whose loci are on the same chromosome, therefore don't independently assort. They tend to be inherited together and don't follow normal Mendelian inheritance. The phenotypic ratio will be more closely aligned to a monohybrid cross, as the two genes are inherited from a single unit. So when Morgan crossbred the red eyes with the white eye mutants, he discovered a sex bias. He discovered that all female offsprings of a red-eyed male were red-eyed. However, all males from a white-eyed female were also white-eyed. So this differs from Mendelian inheritance as it is based on crossover frequencies, that the genes are located on a specific chromosome rather than being independently assorted by the Mendelian ratios. So now here I have a quiz. So the first question is, what was the mutation that helped Morgan perform his research in Drosophila inheritance? The second one is, why is Morgan research important in genetics? So what did this suggest? How many generations did it take until the mutation appeared again in the Drosophila? How did distance of two gene loci have an impact in the phenotype of the Drosophila? What is the main difference between Mendelian inheritance and gene linkage maps? And then I have a word search with all the words that you need to find. And then here are my references. Thank you.